So kiddos, this is cell division, and we're going to be talking about cells when they get specific signals, right? And that could be from human growth hormone, or it could be some sort of, some paracrine signaling molecule from a wound that says to the receptors of that cell, hey, it's time to divide, right? So we're going to talk about that process. In a previous lecture, I talked about how the surface area to volume cannot handle a large cell unless there's a massive increase in surface area due to invagination of the cell membrane, right? And that's because the cell membrane has this incredible function of taking in energy molecules and releasing waste from the cell. And it's not able to do that efficiently if the volume is too large for the surface area to effectively do so. But there are some other reasons that cells need to divide. Cells need to divide for reproduction. So if it's a single-celled organism, that is their only way to reproduce is to actually divide. For some cells, it's going to be for growth and development. And for some cells, it's going to want to divide due to tissue renewal. So how do they know when they're going to divide? How does that process work? That's what we're going to talk about. So it's our old friend, the cell cycle. Yay. Okay, so let's talk about what's going on here. So first of all, you've got the cell and it starts to grow. So it's gotten some sort of signaling molecule where it's going to start to grow. It's going to start using its smooth endoplasmic reticulum to generate some cellular membrane. It's going to start to double up on its membranous systems. It's going to increase the number of ribosomes it has. It's going to increase the number of cytoplasm, or the amount of cytoplasm it has, until it gets to this nice, yummy, full size, right? And at that point, it might just stop there and just do its job for a while. It's grown up now. It's doubled everything it needs to double to function at its optimum capability, right? Well, let's say though your body says, no, I need you to divide now. So the first thing that's going to do before it does anything else, before it increases cytoplasm or anything, it's going to dedicate all of its energy to synthesizing a new set of blueprints. So the nucleus inside of the cell holds this set of chromosomes, which are the blueprints for everything to build the entire organism. So if it's one cell, then it's going to be the blueprints to build one cell. But if it's a multicellular organism like me, I have the instructions on how to make my eyeball in the tip of my toe cells, right? And so there's an entire blueprint there. That blueprint is so incredibly important that the cell needs to make sure that it gets that done first. So it goes through S phase and synthesizes a new set of DNA. As we know, that's your blueprint or your inheritance information. That's everything you got from your parents. Then, and only then, we'll go into the second growth phase. The G doesn't actually stand for growth. It stands for gap phase, but I think that's kind of stupid. So I was in my head, so growth phase, right? So it's going to, once again, it's going to increase the cytoplasm. It's going to tell the mitochondria and chloroplasts, if it has them, hey, you guys need to double. So it's going to send them signaling molecules so that they can reproduce themselves. It's going to increase the membranes. It's going to increase the number of proteins. It's going to increase the ribosomes. So it's a nice, chunky, fat cell, right? About as big as it could be, efficient with its surface area to volume ratio. And then it's going to go through mitosis. Now, once again, the blueprint is so important that it is going to divide or focus on dividing the nucleus first. So mitosis is actually the division of the nucleus, whereas cytokinesis is the division of the cell. So it's going to work on dividing the nucleus, and only when it's pretty much done and it's just the workers are going home, only then will the cell actually divide its cytoplasm and its organelles and separate into two new cells. And then guess what? That new cell, it's going to be kind of small. It's going to start making some cytoplasm, plumping up a little bit, making some endomembrane system, making some extra ribosomes, getting up to that full mature size. At that point, if the hormones or the signaling molecule is still flushed through the body, saying you need to divide, so if this is human growth hormone, then it'll start to go through the whole process again. If it doesn't have a signaling molecule telling it to divide, it's just going to chill out. And we're going to say that it goes into a G0 phase. So there's this little phase where it can go out over here where it just chills out and does its job. In some parts of the body, once you hit maturity, like that's where most of the cells are. In some parts of the body, like your cheek cells that are always reproducing, they're very often going through this cell cycle. Oh, by the way, this whole part, G1S and G2, where it's not dividing, that's called interphase. Okay, and mitosis and cytokinesis are called the mitotic phase. So before we go too much further, I need to clarify a little bit about DNA, chromosomes, chromatin. There are a lot of terms that sound alike, and I want to get through some of this vocabulary. So first of all, the DNA for an organism is found within the nucleus, inside of this nuclear envelope. In general, the DNA is coiled into what we call chromatin, which is loosely coiled up, but it's nice and loose so that the cell can still get to the DNA to read the DNA at any time it wants, but slightly organized as it's gently wrapped around these histones so it can easily access that information without it getting like all tangled up or anything, right? So this right here would be, lost my mouse, this is one strand of DNA, this is another strand of DNA, and this is another strand of DNA. These are each individual chromosomes. So these are each look like this. In the S phase of the cell cycle, they duplicate. So if this was chromosome 1, I would still though refer to this as chromosome 1. It's just chromosome 1 in its duplicated state, ready for division. Once it divides the two separate cells, this is chromosome 1 in this daughter cell, and this is chromosome 1 in this daughter cell. So it gets a little bit confusing, but we have some specific names to help clarify this for you. So this is chromosome 1, okay? And then in S phase, it duplicates. We're still calling this a single chromosome, but we separate it out into sister chromatids. So here's one chromatid, and here's the other chromatid. And those two chromatids are actually genetically identical because they're both technically chromosome 1. I know it's weird. Chromosome 1, chromosome 1, chromosome 1, chromosome 1. This is the parent cell, chromosome 1. It's duplicated chromosome 1 so that it can give one chromosome 1 to each of its daughter cells. Okay, that is this right here. So this is still chromosome 1, one sister chromatid, another sister chromatid joined together at the centromere. All right, so the cell is ready to divide. First of all, we've got our nucleus. It's protecting the chromatin, right? Uncoiled. And then the cell is given the signal like, oh, you have to divide. So it starts to go into the S phase of the cell cycle. And at that point, the chromosomes are going to, or the chromosomes within the chromatin are going to duplicate themselves, right? So they're going to duplicate themselves, and then the cell is going to enter into prophase. In prophase, you're going to have an aster start to form as the centrioles start to shoot out little spindle fibers. The centrioles have congregated. The nuclear membrane is going to start to dissolve. And by the end of prophase, also known as prometaphase, the spindle fibers are attaching at the centromere of the chromosomes, which consist of two sister chromatids, and it's shoving them towards the middle. At this point, the nuclear membrane is almost a fragment, and the different asters have gone to their opposite poles. Okay. In metaphase, the chromosomes line up in the middle. Meta means middle, so the chromosomes are lined up in the middle along the metaphase plate. And in anaphase, I say in anaphase, they make A's because they get pulled apart from each other. And in telophase, you can start to tell those different nuclei apart because the nuclear membrane is going to start to reform. The aster is going to start to break down. The spindle fibers have mostly disappeared. At this point, that's when cytokinesis typically occurs. In animal cells, you're going to form a cleavage furrow, and in plant cells, it's going to form a cell plate in between. I refer to this process as the PPMAT process, right? So you've got I, interphase, PP, prophase, prometaphase, MAT, metaphase, anaphase, telophase. IPPMAT. I don't know, it sounds like something a little puppy would say, right? None of that should be new to you or particularly interesting. Okay, interesting, absolutely. But I did want to introduce this idea of these microtubules shooting out of the aster. So this is an animal cell. So the microtubules are shooting out of the aster. I also want you to remember that along the metaphase plate, they're not like perfectly in one single line. They're all along this plate, along the plane, right? And so you've got these microtubules attached to the centromeres. Now, we used to think that the microtubules were disappeared here, and it kind of pulled them like a rope back to the other side of the cell. But what we now know is that the kinetochores actually munch on these guys. And so they break them down, and because they break them down, they slowly get pulled up, kind of more like the kinetochores are slurping a noodle and heading this way instead of these guys pulling the chromosomes in like a rope. So once the cell is almost done with mitosis and telophase, then the cell is going to start to go through cell cleavage or cytokinesis, and it's going to cause a cleavage furrow here, or in plant cells, a cell plate is going to start to develop. So the way that we describe cell division is kind of like, oh, this is the general world. But as you know, there are always really weird exceptions. So if you looked at some types of eukaryotic cells, they're going to have all sorts of weird things that they do as part of their cell division process, and that's fine. You just need to know the generalities of it. But I did want you to understand that prokaryotes, because they don't have a nucleus, they just have this like region of genetic material called a nucleoid. Their cell division is just a little bit different. So what happens is they, like eukaryotic cells, they double their DNA, but then this 2 copies of origin right here become attached to different parts of the cell membrane, and as the cell separates, it kind of just like whoop, unzips the 2 circles that were connected together from that the DNA replication process. So once that DNA is replicated, it's all stuck together, then it just attaches it to different sides of the cell and then pulls it apart. And that is called binary fission. So how is this process regulated? Well, I already talked to you a little bit about how cells can go into this G0 phase. Like if they're told that they're cool, they're just maintaining homeostasis and they don't need to divide, then they'll just go into this G0 phase. But then they'll get a signal, right? And that signal will tell them they need to move past this G1 checkpoint. So at the G1 checkpoint, they can either move forward and divide, or they can hang out and chill, okay? So if they're told that they need to go through that G1 checkpoint, then they're going to go through the S phase, and they're going to double everything. And there's going to be the G2 checkpoint. The G2 checkpoint, the cell kind of does this quick self-analysis and says, hey, am I big enough? Do I have enough organelles? And did I synthesize my DNA correctly? If it did all of those things, then it'll go through the G2 checkpoint. It'll go through mitosis. At that point, it's going to double check its DNA. As long as there are no major gaps in the 2 new sets of DNA, then it'll go into cytokinesis. If it does not make it through this process, the cell will kill itself, go through apoptosis, or it'll go through cytokinesis and generate 2 new cells, which will then each both start the cell cycle. We found there's a couple of chemicals that are associated with the progression of the cell cycle, and I wanted you to be familiar with those. So we've got cyclin-dependent kinases. If you remember, kinases phosphorylate things and typically start like phosphorylation cascades and amplify signals and make stuff happen, right? So I've got the cyclin-dependent kinase, and when it is bound with cyclin, it comes together to form MPF. And MPF is then going to cause the mitotic division, right? And then the mitotic division is going to break apart the cyclin, which is going to degrade off the cyclin-dependent kinase, deactivating it, and it's going to go back into the G1. So if you can see this right here, we've got MPS and cyclin concentration are low, so we're in G1. S, G2, oh, gotta go through mitosis. So now the cyclin-dependent kinase gets activated and goes through mitosis, and then, oh, it's all degraded. Okay, so you can see that process. I'm sorry, kiddos, I know this is a long lecture, but let's talk a little bit about what happens if that cell cycle, the checkpoints become mutated or don't work correctly, or for whatever reason, going back, this MPF doesn't degrade back into the inactive form of cyclin-dependent kinase. What if it's just told to keep going and keep going, keep going? What if some of those receptor proteins in the cell membrane get somehow jammed, so they hold on to that ligand molecule that told it to reproduce, but it's no longer flush in the system. There's so many different mutations that could occur that could just keep this quick reproduction going. Okay, so if that happens, then that's what we call cancer, right? So when we have cells that are unregulated, that they are reproducing really quickly, oftentimes they keep reproducing so fast that they're accumulating more and more and more mutations. They're turning weirder and weirder and weirder to a point where sometimes they lose all of those proteins in their cell membrane that tell them where they're supposed to be and help them communicate with other cells around them, that tell those cytotoxic T cells like, hey, I'm sick, I don't know what's going on. If those protein markers break, right, if that happens, then that cancerous cell can move from its organ of origin and then either link someplace else or just start growing randomly inside of your body, get caught in your lymph, get caught in your lungs, can get caught all sorts of places, and then start to grow out of control there. And that's when cancer gets really bad, because once it spreads to your whole body, then it becomes much more difficult for us to cure it. So we're going to talk about that a little bit more in class. I wanted to cut this lecture short now. I hope that you can start to think about how you could tie this to cell communication, tie it to amino acid sequence, tie it to nucleotide sequence maybe. Can you tie this most definitely to the processes of mitosis? And on that note, I'm going to end this lecture.